20 seconds. 10, 9, 8, 7, 6, 5, 4, 3.
This egg contains all the information and all the chemicals needed to make a chick. So let's have a look at it, if I'm clever enough to open it. There we are. Perhaps you could put that down, Bill. There we see the familiar egg with its yolk in the middle, surrounded by the white. Now the yolk is mostly fat and protein, and those are the materials that are going to be used in building up the chick. And around it, the white, that's mostly water, but also some protein. We saw an enzyme which is present in the egg white a little earlier on. But also in there is a tiny speck attached to the yolk, which you can see only with difficulty, which has got in some way the beginnings of the chick in it, because we can watch how that chick develops. If we follow the pictures taken by Oxford Scientific Films, then in the slides we can see what this egg looks like. Here's the egg to begin with a rather beautiful circular hole cut in it so that you can see the yolk. And here are two white spots that are simply the lights illuminating the egg when the photograph was taken. But here's another little white spot, and that's where the chick is going to begin. Now, if you take a photograph at close quarters a little less than 2 days afterwards, you see in the next slide that something rather remarkable is happening, that a chick is beginning to develop, and you can see here the beginnings of a backbone with all these bodies, which are protein at this stage, developing. And if we go on to 2.5 days or so in the next slide, you can see that quite a considerable structure is developed. This is going to be the head of the chicken, and here you can see an eye beginning to develop. And if we go on to about 4 days in the next slide, you can see over on the right the head with the various parts of the brain developing and places where the eyes are going to be. And if we go on for about a week, then here we have a really quite marked eye. It's really a bit later than the week, I think, nearer 2 weeks. And in addition to the eye that's developed, here we have the beginnings of a beak, and this little white speck here is an egg tooth. That's going to be used by the chick to get out of the shell after about 3 weeks. And that's shown in the next slide. Here, after 3 weeks, we have the chick coming out of the eggshell. So all of the information for that was already inside the egg, and after 3 weeks, these are the little creatures that come out, these little chicks. So starting with the egg, in 3 weeks, we get to these little chicks. How does that happen? It's obvious from those slides that there wasn't a tiny chick inside that simply got bigger. It was something more complicated than that. If we look rather closely at chicks or ourselves, it turns out that we are made of very small cells. Here's a drawing of a cell. This is actually a nerve cell. In this drawing, you see a rather complicated shape because nerve cells have to be in touch with a lot of other cells and they're making contacts with them. And this isn't a complete drawing because if we went on in this direction, we might go on for a long way. Actually, in your body, we might go on for a yard to communicate through this nerve cell. Also notice inside there are structures which can be seen, and here is represented what's known as the nucleus of the cell, which we shall be thinking about a good deal. But let's not just stay with drawings of cells. Let's use the microscope over here and have a look at some real cells. Now in the microscope we have some lung cells. There are some lung cells, and you can see the big white patch in the middle is an air passage through the lung, and that's surrounded by cells, and you can see the little black spots in them, which are the cell nuclei, and notice their shape. This is how lungs are made up. But do you know where lungs are? Let's have a look where lungs are. Here we have Val Penny, looking very attractive on the outside. Are you sitting comfortably, Val? Then I wonder whether you could show us your insides. Here you see, and in the audience, if you look at the monitor, you see that we can have a look at Val's lungs. I hope she doesn't mind if I try to take one of them out. Let's try to take one out. Here's part of Val's lung, and I can take that out and turn it around. And there you see the sort of porous structure with air channels running through it that we saw in the slide. This is where the air comes in to collect, to bring oxygen into the lungs, which is then picked up by the blood in the way that Dr. Perutz will be telling us about in the next lecture. But we can take out, if Val's still feeling all right, we can take out...
You've got very cold hands.
We can take out another piece of lung, and that will reveal in the middle there the heart. So let's have a look at some heart cells. Under the microscope, there we have some heart cells. Now, these are muscle cells of a slightly different kind than the ones we looked at in an earlier lecture, but you see that they're stretched out mostly in one direction and forming these muscle fibers within the heart. There's the heart, a good sort of heart shape, and I can take that out. And now we are exposing the diaphragm, and underneath that, the liver. Here's the liver. We looked at liver in an earlier lecture because we were looking at enzymes in liver. I can take that out, and there it is, rather a lovely big piece of liver. And under the microscope, we can look at some liver cells. And here again, you can see them packing closely together, but of a different shape. If you look at them closely than the other cells we looked at, again, you can see the nuclei in them. Well, I think perhaps it might be better to put Val back together again now. So let's put the liver back. And then, having got that back in, I think that looks in the right place. Let's put the heart back in. There we are. Do you think that's in the right place, Val? Yes.
Yes.
Right. And now we'll put in the top of the right-hand lung. All right. And now the top of the left-hand lung. Let's make sure that's all right. Are they comfortably in place there? Good. Thanks.
Fine.
Well, perhaps that's as far as we can go, looking at where these organs are and the cells that they're made of. Thank you very much, Val. Perhaps you'd like to cover that up again. Thank you very much indeed. Well, thank you very much, Val Penny. That goes to show what can be done with a couple of cameras and Val Penny and Val's other self. And I think perhaps Bill had better take Val's other self out as well. So it seems that we're all made of cells of different sizes and shapes in different parts of the body. How then does that all begin? It all begins from one cell. And one cell divides and forms two cells, and those two cells divide to form 4 cells, and then we get 8 cells and 16, and so on. And as they divide in this way, when you get to very large numbers, the cells begin to change in character. So some become liver cells, and some heart cells, and some muscle cells, and so on. It's clear, though, that cell division is a very important process. So let's look at that in a rather simple organism, in fact in a plant. On the next slide, we see a number of crocus cells at different stages of cell division. Down at the bottom here, we have a cell with the nucleus showing up as a rather grey diffuse body, but beginning to develop some structure. If then we look up at this one, the nucleus has become even more structured, and you can see some thread-like objects which take up coloured stains beginning to appear in it. And in this one, these thread-like objects have become thicker and they are easier to count. There are actually 6 of them. And when we get to this cell down here, these thread-like objects have become rather compact and fat. They are called chromosomes. There are 6 of them here, 1, 2, 3, 4, 5, 6. And they are just in a state to divide in half along their lengths so that each one will become 2. And then these 2 sets get pulled to the 2 ends of the cell, as you see in this one. Now these 2 new sets of 6 chromosomes, in the cells in your body you have 46, not 6. These sets now become the nuclei of 2 new cells. And over here you can just about see the nucleus of one daughter cell. There's the nucleus of another daughter cell, and there's a membrane forming between the 2 so that this is becoming 2 cells. So something's clear about cell division. It's clear that when cells divide, important things go on in the nucleus. These thread-like bodies, which we call chromosomes, are copied in some way so that they split into 2, and 2 new sets go to 1 to each of the 2 daughter cells. So we have to think about chromosomes. What are they made of? Well, they're made of a new substance that we haven't met before, which is called nucleic acid. There's also some protein there, but I'm not going to go into that. Let's suppose that they're mainly, as they are, nucleic acid. What is nucleic acid? Well, that's a rather complicated chemical, and this drawing, I think, really does make it seem rather complicated. But it's not as complicated as all that, because it's another long-chain polymer molecule. And if we look at it slowly, these green 5-sided rings here, they are sugar molecules, a particular sort of sugar molecule. And here's another one, and another one, and another one. And these sugar molecules are connected together through phosphorus atoms. There's a phosphorus, there's a phosphorus, there's a phosphorus. So we have sugar, phosphorus, sugar, phosphorus, sugar, phosphorus, sugar, and so on for a very long way. So that's a very long polymer, made of sugar and phosphorus, like that. But that isn't the end of the story. Over on this side, you see that attached to the sugar, there are also other groups, and there are other groups of 4 different kinds. This drawing just shows 4 units so that I can show each of these 4 different kinds. This one we are going to call A for short, this we'll call C, this one we call G for short, this we'll call T. And notice that A and G are both made of a 5-membered ring and a 6-membered ring joined together, and C and T are made of a 6-membered ring with other groups of atoms attached to them. So hold on to A, C, G, and T, and to this sugar phosphate chain, and now we'll try to make it look simpler. Here's a rather simpler drawing. Here's the sugar, phosphorus again on this side, but now these side groups, which I shall call bases. If I start talking about bases, I'm talking about these groups A, C, G, and T. And here I've put in the little charges that occur on them. These are polar molecules. They are carrying little electric charges. And A is carrying a plus and a minus, and C is carrying a plus and a minus and a minus, and G is carrying a minus, and a plus, and T is carrying a plus. And you could imagine that because of this little arrangement of charges, these little molecules might fit together like jigsaw puzzle pieces. And that indeed was one of the inspirations of Watson and Crick in 1953 when they were working on this structure of nucleic acid. This is the structure of DNA for short. We'll come back to what DNA might mean in a minute. What they realized is that these bases, A, C, G, and T, might fit together. And indeed they do, and they fit together in a rather neat way, so that A, if I turn it round to the other direction from what it was before, fits together with T, making plus, minus, minus, plus interactions. And G fits together with C, making minus, plus, plus, minus, plus, minus interactions. And the very nice thing about this is that these pairs, and I shall call them base pairs sometimes, these pairs are geometrically very much the same, because the distance from this position, where this is attached to the sugar, to there, where A is attached to the sugar, is exactly the same as the distance from there, where C is attached to the sugar, to there, where G is attached to the sugar. So in some ways, these base pairs look identical, although the individuals, A, T, C, and G, are really rather different from one another. That's another example of jigsaw puzzle pieces fitting together. Now, where can we go from there? This is a polymer molecule, and we talked about polymers before. What kind of three-dimensional structure would we expect a polymer molecule to take up? Well, we decided earlier we'd expect it to take up the form of a helix, a sort of spiral spring structure like this. How do we find out how the atoms are arranged in a structure like this? We have to use a microscope with X-rays, and if we haven't got crystals to look at, then we want to look at a helix like this and see how this will scatter the X-rays and see if we can deduce from that what the structure must be. So we have a problem. How do we find out how a structure like this would scatter X-rays? Well, we can build that up, but first of all, let's look at this in a particular direction. If I look at it in that direction, then you can see, looked at in that direction, perpendicular to the axis, this helix looks like a wave. It's just a wave going along. So let's think about a wave like that on the overhead projector and how that would be likely to scatter X-rays. And we can do that by thinking about how a similar structure would scatter light using the laser beam that we used before. If we look at this wave now, you can see that you can almost pretend that there are straight lines there and there. So we can represent this as a set of lines. Now, we've looked earlier at the way in which a pattern of straight lines on a glass plate would scatter light, and if Bill puts such a pattern of lines in the grating, you can see that it's scattered. Here's the central beam, and here are beams coming out on either side in a line which is at right angles to these lines in the wave. Well, it's clear that I can put in some more lines in this wave like that, which we can think of scattering light in another direction. That's not fitting very well, is it? But there we are. There are some lines like that. And if Bill puts in the pattern of light, of lines in the laser beam, here's the central beam still, and now we have another line of scattered light beams running along a line perpendicular to the second set of lines on the wave. Well, so far I've been talking about just a continuous wave, but we know that it's made up of atoms and groups of atoms which are likely to be at regular intervals along the wave. So I can put in another set of lines at regular intervals along the axis of the wave like that. And if Bill puts in a set of lines in front of the laser, these lines are closer together than the other lines, so the first wave of light wave scattered by them is further away from the center, and it comes up just there, you see. So the total pattern, which I think is rather beautiful, is across that way and this way, and then on the horizontal line there are these spots which arise from the units going along the wave. Now, thanks very much, Bill. Let's remind ourselves of that by looking at a diagram of it. Here's a diagram which shows again what we've just seen. Here we have the wave, and here is this cross where the spots on the cross arise from the light scattered from those lines and those lines. And along the horizontal, there's a spot there and another one there, and that arises from the objects which are at closer intervals along the wave. The closer they are together, the further from the center the spot is. So that, in a short time, is the theory of how a helix would scatter X-rays. When Watson and Crick and Wilkins and others were studying DNA back in 1953, imagine the excitement when they looked at the way in which a DNA fiber scatters X-rays, and that's shown on the slide. Here you see, immediately recognizable, the cross in the center, which is telling them about the pitch of the helix, and this black spot along the horizontal, which is telling them how far apart the objects are along the helix. It turns out, from the dimensions, you just have to measure this with a ruler, that the objects along the helix, there are 10 of those in one turn of the helix. Then, of course, Watson and Crick were able to put this information together with their idea about base pairs, A plus T, having the same size as G plus C, and they built up the famous double helical model of DNA, which you see here. Here is the double helix of DNA. Here are the objects at right angles to the helix axis, and these are the base pairs, AT and GC base pairs. And here we have the two helices, because it's a double helix. Here is the sugar phosphate chain on this side, and a sugar phosphate chain on this side, these chains running in opposite directions, and linked together through these base pairs, so that A on this helix fits onto T on this helix, or G on this helix fits onto C on this helix, and so on. A rather beautiful structure coming out of that rather funny photograph. If we look at a different model, this model is made of components which show that atoms really fill space rather than being little balls held together with rods. They fill space, and if I turn this, you can see what a beautiful sort of corkscrew it is. So that's the three-dimensional structure of DNA, the famous double helix. Does that tell us anything about how the chromosomes, which are made of DNA, might be copied exactly in going from the parent cell to two daughter cells? Well, Watson and Crick immediately saw that it did. And if we look at a rather simpler model of DNA, here we have a DNA double helix, then we can see what the inspiration was. Here's a not very helical model, but it shows the base pairs, AT, white and blue, GC, green and black, though I'm not going to try to remember those colours. What Watson and Crick saw was that if I pull this apart, breaking these base pairs, the last one's always difficult, there it comes, breaking these pairs, I now have two separate strands. And these strands could act as strands on which new double helices are built up. Suppose we have the various components about in the solution, lots of A and T and G and C about in the solution, then they could come and attach themselves to these separated strands of DNA, and we might get a copy. Now let's see if we can do that. What I want are some volunteers. Here are the things. Let's have some volunteers. You, and the boy behind you, and yes, I think you'll do fine. And that little, now we want another little girl, really, don't we? Yes. How about you? You come and do this, all right? And I think we could maybe do with a couple more. Yes, you'll do fine, and you. Come and join these teams. Now, the largest of you, would you like to come over on this side? All right? Could you hold on to that? And you hold on to it on that side, and you join this team, and you this one. Now then, here are the parts, and here are your parts. See if you can build up some new DNA molecules. Remembering, and this is the key thing to keep remembering, that G goes with C and A goes with T. Right? Yes, they're coming on rather well. Is that one complete?
Yes.
The one on the left is complete, right? Stand back. How about the one on the right? Very good. This was a very good team. There's one complete one. And that's nearly there. A white one to come there. Very good. Thank you very much indeed. There we have two complete DNA molecules. Thank you. So these are now identical copies of the one we started with. But remember one thing which is very important, that although it looks as though the DNA did it all by itself, together with these components, there would have had to be enzymes, proteins involved, making these chemical connections between the components. So it needed proteins to copy the DNA. Never mind, we've got two identical copies, and that is a perfectly reasonable plan for how the DNA in the chromosomes is copied so that the parent cell can make identical copies to go to both daughter cells, and they go on doing the same. So the master plan, if it's written somehow in DNA, can be sent on to further generations of cells, and indeed to further generations of people. Well, it's all very well to have a master plan, but nothing very much happens if you keep on copying a master plan. Something has got to happen. In a factory, that would be the master drawing in the drawing office, and blueprints would have to be made of that to be sent out into the factory for the engineers to use in manufacturing their product. So the DNA is the master plan in the nucleus of the cell. We want a blueprint which can go out into the rest of the cell to get things done. So what's the blueprint going to be? The blueprint is made of another sort of nucleic acid, which is very closely related to DNA. So we'll go back to this rather fearsome-looking drawing. When I was talking about the sugars, these green painted rings, I said they were sugars. I didn't say what their names were. DNA means deoxyribose nucleic acid. And deoxy means that it's missing an oxygen, short of an oxygen. If I put in the oxygen that was missing just there, I go from deoxyribose to ribose. So if I put that in all of these places, I'm changing DNA to something else. I'm changing it to ribose nucleic acid, or RNA for short. But I've not quite completed the change. There's one other change. This group T down here loses carbon, and that group is just replaced by a hydrogen, and that changes the chemical nature of this T into something that we call U. So whenever I've said T before when talking about DNA, we've now got to think about U. But that doesn't affect the way in which A fits onto it. A used to fit onto T, it fits onto U in exactly the same way. So the base pairs we have to think about are now AU and GC rather than AT and GC. Otherwise, RNA is like DNA. And that's the material of the blueprint. You can make RNA on DNA in just the same way. If I pulled those helices apart and you had the components now for RNA, you could build that up to form an RNA string, though it would involve different enzymes. And the RNA comes out of the cell nucleus into the rest of the cell. What does it do there? Well, the things that really make things happen in cells are proteins. They're the machine tools of the cell. Somehow, we've got to make protein. And nothing, the master plan is useless unless the master plan can somehow instruct machinery in the cell how to make protein. So now we've got a coding problem. Out of the nucleus of the cell comes a long string molecule of RNA, and we can abbreviate the formula like this. It looked very complicated on the diagram, but for short, it's simply something like this, a string of letters involving A, U, G, and C in what would look like a quite random order if we wrote out the formula. That's the structure of the blueprint, the RNA. The protein, on the other hand, we know is written in a different way. This is written in brief like this, in a string of 20 different letters. So we have a problem. We have to go from a message written in 4 different letters in the RNA to a message written in 20 different letters in proteins. That's the coding problem. How do we solve it? Well, what sort of codes could we think of? We could think of words of different length representing something else. That is, well, the shortest word is a 1-letter word. If we have a 1-letter word written in the RNA language, in the RNA alphabet, then we've only got 4 possible 1-letter words. It's either A, U, C, or G. And if each of those represents one protein component, those protein components are known as amino acids, incidentally, and I might start using that now, then we could only code for 4 different protein components. But there are 20 of them, so that isn't enough. What if we had 2-letter words? Well, there's a problem for you. If we have samples of these letters, A and C and G and U, how many different two-letter words can we make? Let's have a game about that. Let's see who can do this. What we want are, yes, you'll do fine, the boy with your hand up there. And you'll do for another one. You've been out before, have you? Yes. Let's have somebody fresh. Let's have you in the front. And yes, let's have you as well. And we want another little girl somewhere who can get out. Where's a girl who can get out? Yes, let's have you. Fine. Come on, you come on down. Now come around this side. Let's have two boys over here, right, and the two girls over there. Now here are boards with lots of pins on them, and there are supplies of these letters. What we want is all the two-letter words you can write with those letters, where, for example, AU is a different word from UA. It's permutations you're after. Now, starting now, how many can you do? Well, they're doing very well, aren't they? There seems to be some sort of regular pattern building up over there. So far we've got 2, 4, 6, 7, 8, 9, 10, 14 here, but how many doubles have we got in that? No, that looks all right. What else have you got? Anybody nearly finished, do you think? You think you've finished over there? The girls think they've finished. Let's have a look. Now, let's look at that. Now, they've been very clever about this. They started by putting down A's in the first place, and then U's in the first place, and G's in the first place, and then C's in the first place. And then by each A, there's C-U-G-A, and by each U, there's A-G-U-C, and by there, and by each G, there's G. Well, that's certainly right. And how many are there altogether? 16. Now, how many did you get? 3, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So yours may be right too, but they'd take much longer to check, wouldn't they? Anyway, that was good. Thank you very much indeed. $16, well, that's quite a good number. 16, that's nearly 20. But it isn't quite 20. We need at least 20 because there are 20 components that proteins are made up of, and there may be some full stops and so on, and some start signals. So 16 isn't enough. Suppose we have three-letter words. How many three-letter words can we make out of 4 different letters? Any volunteers? All right.
64.
64. Yes, that's quite right. 64 different three-letter words. Well, that's certainly enough, and it turns out that that's just the way it is. Here you see on this drawing, we have the code which relates the structure of RNA to the structure of proteins. So, for example, if we look at a protein component W there, then the first letter in the three-letter word is written down over here. That would be U. And the second letter is written here. That would be G. And the third letter is written there. That's G as well. So the word for W is U-G-G. Right? Let's try another one. Let's try Q there. The word for that would be C-A-G or C-A-A, because there are several different words sometimes that code for the same protein unit. Now, let me see what you could do. What about M? Can anybody tell me what the code is for M? Right? You?
AUG.
AUG, that's right. I've lost it, UG would code for M. What about G down at the bottom there? Right?
GGG.
G, G, G. Splendid. So that's how the code works. Three-letter words in the RNA tell us a protein component. We can translate then from the structure of RNA to the structure of protein. Well, that's all right. We can translate it, but we need apparatus to make the protein. How are we going to do that? Well, that's a little tricky too. We need some sort of enigma machine or some kind of conveyor belt, some sort of production line on which to make protein, guided somehow by the message written in the RNA. Now here we have behind me a representation of this machine. Here's the production line. And in it, we have a slot where the message is going to come. I can wind that on, and you'll see that the message in RNA is going to come in there. But this is made, in fact, of a lot of proteins. Something like 60 proteins are used to make this apparatus in the cell, in all of your cells. And about 2/3 of it is actually made of another kind of RNA. So it's a mixture of RNA and protein. That's RNA in addition to this piece of RNA. There's RNA making up the works as well. And of course, among the proteins, there are enzymes. So somehow we shall have to fit some enzymes into this, and we shall need one in particular. So Sarah, I think, had better come and be an enzyme. If you like to get into your right position on the production line, then you can be an enzyme. Fine. Now, what we're going to do on this machine is manufacture protein according to the recipe written in the RNA structure. So how do we do that? Well, we shall need some more molecules to carry the parts to the production line and do other jobs. Some carrier molecules. What are they going to be made of? Well, the carrier molecules are made of RNA again. And here is a structure of such a carrier molecule. It's not the three-dimensional structure, although that is now known. It's a chain of RNA shown rather diagrammatically here. But down at that end, we have 3 of these bases. And their job is to recognize the 3-letter word. Now, how would you set about recognizing this 3-letter word, AUG? If you were nucleic acid parts, you would say, aha, A always goes with U, and U always goes with A, and G always goes with C, and so on. So let's be sure that we remember that. That's a very important thing to remember. I'm going to hold this up to begin with, and then I think what I'll do is to give it to somebody who can hold it up to remind us all the time. So let me now give that to someone in the audience. I think you're in a good position. If you sit down and just hold it in front of you, something like that, then apart from the people behind you, we can keep reminding ourselves that G goes with C and A goes with U. So how would I recognize this? I want a molecule which has got U at this end and has got A in the middle and C at that end. U, A, C, to recognize that. All right, well, this molecule that we were looking at over here, down at the end there, has got U, A, C. That's all right. Then this molecule could come along and fit on to the production line. But it needs to be carrying something with it. It needs to carry with it the protein part for which the code is AUG. And if Sarah would move a little bit to her left over there, then we could see, you need to move a bit over there, we could see that that would be AUG codes for M. Now, M, that's a funny one. M means not only one particular protein component, it also means the start here. That's a bit complicated, and we won't go into that very far. It means start, let's say, and it's carrying protein component M. And this molecule will do that by putting M on there. There's now the protein component sitting on the carrier molecule that corresponds to this anti-code at the bottom. The anti-code is going to recognize the code. That seems rather complicated, but let's try that out on a living model. I'd better have that one. I'm going to be the molecule we've been looking at, the one that starts. So I'm going to recognize AUG. So I'll put around my neck this poster which says UAC on it. Here I am saying UAC, so that I will recognize AUG. Now we need some other carrier molecules too, so I want some volunteers for carrier molecules. I think we'll have the one there who was the winner in the other scheme. These are the carrier molecules. Would you like to put that on? And then let's have these two boys here. There's one for you, if you like to come out to the front. And another one. You're a carrier molecule. And then we want another girl. I think we'll have you. That's right, yes. Let's have you. So here we are. We've got 4 carrier molecules. They are going to carry the protein parts to this production line. But how do they get the protein parts? Well, we need some enzymes. We need a protein which can recognize the carrier molecule and attach to it the correct protein part. All right? So here are protein parts. I'll take one lot. And I want some enzymes now, and they'd be better be sitting in good places. Yes? Let's, whoops. Here you are. Here are your protein parts. Put them on the seat by you. And here is a label which you hang around your neck, right? And you're the enzyme for one of these carrier molecules. Yes, I think you'd better be another one. And you'd better be another one, whether you would or not. And you'd better be another one, all right? And there are your supplies of protein parts. Now we are ready to make some protein. What we have to do is, first of all, the carrier molecules have got to identify the enzymes which can load them up with the right protein part. The carrier molecules are labeled with the anticodes, and the enzymes are also labeled with the anticodes, so that GUG, down here, for example, has got to find the GUG enzyme and collect one protein component. Here's a protein component. This is my starter, right? The enzyme is very exact in how it does these things. I was not using an enzyme just then, but the enzyme has to put this component in the right hand of the carrier molecule with the blue square pointing upwards, right? Now see if the carrier molecules can find their enzymes and get one protein part. Right, now we are ready to begin. So you're all in solution and you're carrying your protein parts. And I am going to approach this assembly line. There I see AUG, so I'll get ready but the message is going to wind on in the assembly line until it's in the right-hand slot as the machine sees it. There we are. Now that's the slot in which the starter signal goes. That's AUG, so I go up and get in position. AUG, that fits onto UAC, so I'm recognizing AUG behind me. AUG, that goes with UAC. You'll remember, looking at the diagram over there, that G goes with C and A goes with U. Now I hold up my protein part, and that attaches to the enzyme up at the back there. Now, notice that in the slot on my left, there is another three-letter word that says GGU. What does that go with? Ryu? CCA. So we want the carrier molecule CCA to fit into that. Here it comes, and it holds up the component, and the enzyme up there will join the components together. When it's done that, it detaches from me, and this carrier molecule can fall off. Right? So I've fallen off. What happens next is, slowly, the message winds on, and at the same time, this carrier molecule moves over with it into this site on the left. Right? If you come a little bit further. Now that's revealed another three-letter word. The three-letter word it's revealed is ACG. What does that go with? ACG goes with UGC. So up comes the carrier molecule for UGC, holds that up where the enzyme can attach it. That joins together. This one releases and comes away. Now this carrier molecule has to find its enzyme again and get loaded up with another protein molecule. The message will wind across this side, that's right, revealing a new word. A bit further, I think. The new word is CAC, so that goes with GUG. So the carrier for GUG comes along, holds up its component, the enzyme attaches it. This one comes away, the message winds on, this moves to the left, and we've got UCA looking for AGU. AGU comes, fits on. This one detaches, comes away, goes back to its enzyme for another component. This one moves over, and we've got ACG again, meaning UGC. So here comes UGC. And let's see if that will now go on more or less automatically. Come on, the next one. Right. Then across. Wait. The next one has to come while you're still holding on. Which is the next one? That's going to be AGU again. Up goes the protein component. That connection is made. You release and come down. You move over to the side. Up comes the next one. A bit further. CAC, so we're back to GUG again. All right. And now the next one. GGU is looking for CCA. Here's CCA. There's the next, and so on. And I think we've perhaps gone about far enough. In practice, of course, the enzymes wouldn't be in one place. I should have got them to come and run around in the middle so that they would have been in solution too. And they wouldn't have had the components in their hands. They would have been in solution as well. We ought to have had a good solution out in the front there with all of these people. But I think they've made some very good protein. Let's look at it. Thank you all very much indeed. Well, we didn't really get terribly far with that piece of protein synthesis. We've only got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 of these protein parts linked together in a long chain, and the chain is hanging from the side of the apparatus up there. And you know that real proteins, like the collagen that we talked about earlier, they would have a long, long chain with as many as 1,000 components in a long line. So we'd really only just begun. In a living cell, another protein, such as the lysozyme that we looked at earlier, which has just 129 of these protein components in the long chain, that would be made in this way in about a minute. That gives you some idea of how rapidly things go on. But so far, what we've got is just a long chain dangling up from the side of this production line. What happens next? That doesn't have any particular three-dimensional shape to act as an enzyme or a piece of muscle or anything. What does it do? All the information is already there for that long chain to fold up and take a particular three-dimensional structure. And that goes back to those soap molecules that we looked at earlier. Let's imagine that that long chain, as indeed it does, has along it some greasy groups and some groups that like to be in water, like this long string here. Here's a long string. We're imagining now that this long string with some greasy groups and some water-loving groups, that this is like a protein chain. When it comes into water, I'll put it into water like that, then it will automatically take up a three-dimensional shape in which the greasy inside is made of the greasy groups, and the surface, which is in contact with water, is made of the water-loving groups, just as they were in this model of lysozyme that we looked at in an earlier lecture, where the blue groups are the water-loving groups and the yellow ones are the greasy groups for the inside. And that isn't just a single protein chain. If it's a more complicated structure like muscle that we were discussing earlier, which is made of a lot of protein molecules like this, the information needed for them to stick together to form long fibers in muscle, that is also built in to the chemical structure. Such a molecule would have greasy patches on it, which would mean that it sticks onto another molecule of the same sort and builds up bigger and bigger structures. That's the principle of self-assembly. Given the chemical structure of the proteins, which was coded in the chemical structure of the RNA, which was coded in the chemical structure of the DNA in the genes, all of this assembly follows. You get folded up chains building up the big structures in our bodies. Now, where can that take us? First of all, wouldn't it be a good idea if we could use this rather marvelous apparatus in a living cell to make the proteins that we need? There's a great need for proteins like insulin, for example. And nowadays, interferon is very much in the news. Wouldn't it be splendid if we could use this machine to make such proteins? We'll think about that later on. But if we go back to the old question, which came first, the chicken or the egg? You've seen these chicks and an egg. Now we have a new question. To make DNA, we needed protein. To make protein, we need nucleic acid. Which came first, the protein or the nucleic acid?